Good morning, everyone. And for those I don't know in the room, my name is David Brown. I'm the president of the Academic Medical Centers of Mass General Brigham. Thrilled to get things started this morning by introducing our president and CEO of Mass General Brigham, Ann Klebanski, for some opening remarks. Ann.
So good morning, everyone. And what a splendid morning this is. Gary, thank you. So it's truly a great pleasure, and it is truly an honor to be here this morning to celebrate Dr. Gary Ruvkun, who has been awarded, for those of you who aren't familiar with this, the 2024 Nobel Prize in Physiology or Medicine. Just saying that. So great. You're going to have to get used to this now. Okay. So Gary is an investigator at Mass General Hospital Department of Molecular Biology and has been at MGH since 1985. 1985. So he has earned this highest achievement for his contributions to the discovery of microRNAs and their role in post-transcriptional gene regulation, and shares this year's prize with long-term collaborator Dr. Victor Ambros from UMass Medical School. So I know you know this, but I'm going to say it. The Nobel Prize is one of, if not the, let's say the most prestigious awards in the world and is science's ultimate honor. This is an incredible achievement that acknowledges Dr. Ruvkun's long and illustrious career and breakthrough research. So research is such a core component of Mass General Brigham, such a core component of its mission, what we all stand for. We take such pride in our legacy and our people. This is the driving force really behind some of science's most incredible discoveries, and these discoveries as they translate into those things that provide better care for all of our patients. So as the largest hospital system research enterprise in the country, we are so uniquely positioned to convene academic medical centers, university-based research, cutting-edge industry partners to advance discovery with local, national, and global impact. Dr. Ruvkun's work is such a vital part of that legacy. In the past 2 decades since his original findings were published, the critical role that microRNAs play in health and disease has become more and more apparent. It takes time, and their therapeutic applications are in clinical trials now for heart disease, cancer, neurodegenerative disease, and so many other ones. So this is the broad implications of how this kind of fundamental research ultimately translates into better health, locally, regionally, nationally, and again, globally. So on behalf of the entire Mass General Brigham community, it is such a great honor to congratulate Dr. Ruvkun on this truly remarkable achievement and thank him for all of his contributions to science, to medicine, and to the health of people. His work extends a legacy of innovation, and it inspires the next generation, so many of whom are here, as they pursue new breakthrough discoveries with, again, global impact. So thank you again for all you've done, for all that this work will accomplish in the future. Thank you. And I'll now turn it back to David for more comments. Thank you.
Thanks, Ann. So I too am just thrilled beyond words to be part of this incredible celebration and recognition for you, Dr. Ruvkun. Wow. We celebrate really with the entire world your incredible achievements, and we celebrate the power of science, the power of brilliant minds, and the commitment across Mass General Brigham to science, the commitment to the 4 pillars of science: basic research, translational research, clinical science, and outcome science. and of course, to the research-infused and research-informed care that is our differentiator. And so today I am thrilled to welcome you as Nobel laureate, Dr. Gary Ruvkun. That has an amazing ring to it, doesn't it? Nobel laureate, Gary Ruvkun. As Anne said, Dr. Ruvkun and his colleagues have pushed the boundaries of science on our understanding of life on this planet and raised questions about life even beyond. Through their contributions to the discovery of tiny molecules of RNA, the so-called microRNAs, which control the activity of critical genes in plants and animals. And now I'm completely out over the tips of my skis, so I won't say anything more about that. Gary, on behalf of all of us across Mass General Brigham, we thank you for your commitment, your passion, your brilliance, your leadership, your mentorship, and we welcome you to the group of Nobel laureates with enormous pride. And again, our heartfelt congratulations. So now it's my pleasure to welcome my friend and colleague, Dr. George Daly, Dean of Harvard Medical School, to the podium for a few remarks.
Oh, yeah. Well, good morning. Yeah, I'm George Daly, the Dean of Harvard Medical School. It is absolutely thrilling. We bask in the reflected glow. We've known you for so many years. We've been expecting this. But let me just get through with some official remarks. I want to say thank you to the MGB media team and for MGB leadership for hosting the conference here, the press conference, which is celebrating the accomplishments of a longstanding Harvard Medical School faculty member, which is why I'm here, but also someone who's been, I did the math, 39 years at Mass General Hospital. I want to point out that Harvard Medical School embraces this curiosity-driven scientific research. The largest number of investigators at Harvard Medical School are embedded within great academic medical institutions like the Mass General Hospital, where their pursuit of understanding of fundamental biological principles inform a deeper insight into disease. And it's this partnership of academia and medicine, referenced by Dr. Kubansky, that is largely responsible for the preeminence of our hospitals and the biotechnology community here in the greater Boston area. Dr. Ruvkun's outstanding body of research is a prime example, and I am absolutely delighted on behalf of the entire Harvard Medical School community that the Nobel Committee has chosen to honor his extraordinary scientific work. As has been referenced, we've been waiting for this accolade to be bestowed on you for quite a long time. And it's been absolutely clear that the importance of Gary and Victor's work has been clear for decades. MicroRNA biology is essential to numerous laboratories across the globe, including my own, where we have turned microRNA regulation and its understanding to a better insight into cancer, diabetes, metabolism, tissue regeneration. Just this morning, I was reminded by my graduate student, Srini Viswanathan, that you and Victor served on his thesis committee. He sent me this. I think you're wearing a slightly better shirt today than you did then. But the theme is still very much the same, you and Victor. I will now get the actual photo and I will be framing it. It'll go up in my office. I'm going to send it for you to sign. But let me just say that the revelation of microRNA regulation, it's absolutely a shining example of how curiosity-driven research can be turned into actionable knowledge that will advance human health for the betterment of humanity. So our congratulations, Gary, on behalf of everyone at Harvard Medical School.
Thank you, Dean Daly. Now I'd like to welcome Marcelo Del Carmen, the president of Mass General and the Mass General Physicians Organization, to the podium for some remarks.
Thank you, David, and good morning, everyone. It is absolutely an honor and a true privilege for me to be here this morning with all of you today, celebrating our esteemed colleague, good friend, and now Nobel laureate, Mass General's very own Dr. Gary Ruvkun. It is because of dedicated, determined, and inquisitive people like Dr. Ruvkun that we've had a long and storied history of excellence in research and biomedical science, starting with the first use of ether as an anesthetic agent in 1846, actually in October of 1846. Our researchers, scientists, and medical teams have taken the spark of an idea and worked tirelessly to take it from bench to bedside. Through trials and tribulations, failures and triumphs, and thanks to the hard work and powerful collaborations of many, together, we are able to offer life-changing and life-saving options for our patients. As a physician, Gary, I want to thank you for all of the breakthrough science that will come from your innovation and your research. And this Nobel laureate accolade will carry impact, will continue to carry impact to the bedside, as both Dr. Klebansky, Dr. Brown, and also Dean Daly alluded to. Just steps from where we are gathered here today, our scientists are hard at work. In fact, many of you are not hard at work because you're here with us this morning at this press conference, but that's okay. We will let you take the break. Like Dr. Ruvkun did for so many years, they too spend their days and nights and weekends pushing the boundaries of science and paving the way for future discoveries, cures, and a better, healthier way of life. Simply put, their dedication and curiosity are benefiting to all of us. Gary, this award is an inspiration to scientists now and for future generations. Thank you for sharing your talents with us and with so many others across the world, and a huge congratulations. Thank you.
Thank you, Dr. Del Carmen. And now I'd just like to welcome Dr. Jeannie Lee, professor and chair of the MGH Department of Molecular Biology, to the podium.
All right. Well, this is an extremely exciting morning for all of us, but I'm sure especially for Gary Ruvkun. So it's a tremendous pleasure for me to be the one to introduce my friend and colleague, Professor Gary Ruvkun, who is this year's winner of the 2024 Nobel Prize in Medicine. So Gary, as it's been mentioned a couple of times already, has been a member of molecular biology for almost 40 years. And it is within the walls of molecular biology that Gary made his seminal discovery about these so-called microRNAs. That's led him to many, many prizes, not just the Nobel today, but also the Lasker, the Breakthrough, you name it, he's won it. Okay. So in 1993, Gary Ruvkun and his friend and colleague, Victor Ambros, who was working actually across the river at Harvard University, and Gary working here, made this amazing discovery, and they published in the fancy journal Cell, describing a completely different way of controlling genes. Okay. And they described that this form of gene regulation is due to a small nucleic acid called a microRNA. And what's amazing about these microRNAs is that they're not just targeting one thing at a time. So as it will become known in later years, they're actually targeting like a whole network of genes. So controlling a lot of different messenger RNAs from many different genes at a time. So it's tiny little RNA with a huge impact on gene expression. But, you know, as it was so often happens in science, the significance of that discovery wasn't immediately appreciated by the community. I mean, it published fancy, it did all these wonderful things, people liked the paper, but the true impact of that discovery wasn't known for many, many years after, probably for a decade or 2 decades even after. I mean, after all, it was discovered in a worm, right? So it was a small microscopic worm called a nematode that buries around in the soil. And it wasn't clear what that had to do with human disease or human health. But fast forward a couple of decades, it's now very well understood that these microRNAs are conserved throughout almost all of biology in plants, as well as in humans. And these microRNAs are important for all kinds of human developmental processes, and they go awry when there's disease, congenital disease, or in all forms of cancer. Okay, so as I said, small RNA, tiny organism with an outsized influence on biology. And so before I turn it over to Gary to actually tell you about the science, I would be remiss if I didn't mention one other person in this audience. Okay, so Victor Ambros can't be here. He's probably celebrating over at UMass today, even as we're speaking, but we have a representative who's actually more than just a representative because she actually performed physically with her own hands the experiments in Victor Ambros's lab. And that is Rhonda Feinbaum. So Rhonda, can you raise your hand? Rhonda's very unassuming. So congratulations also to Rhonda. Now, before I actually turn this over to Gary to make his remarks, I'd just like to say that, again, within the walls of molecular biology, I mean, Gary hasn't just been a great scientist, okay? He's been an amazing colleague. I mean, the best that one can hope for in any department. He's been an excellent mentor to all the young scientists in the department, an inspiration to both young and seasoned investigators in the department, as well as a tireless supporter of doing great science. Okay, so please join me in congratulating Gary Ruvkun, and please, Gary, come up to the podium.
Yeah, it's been a good morning. Thank you. Yeah, this place has been incredible for me. I want to thank MGH for starting the Department of Molecular Biology that recruited a bunch of us 40 years ago. And I think John Potts was really important in this. The endocrinology department at MGH was molecular endocrinology, and they saw that the recombinant DNA revolution was a revolution and was really important in the short run for endocrinology for finding new hormones. And that's how our department started. But almost instantly, our department did a little bait and switch on MGH, sort of a midlife crisis thing where the endocrinology focus switched to everything. And so a bunch of us were recruited in very different fields. And I would guess there was an initial like, what, that's not quite what we bargained for. But the science turned out to be great, all in many different dimensions, and it's worked out great. But I want to thank John Potts for sort of making our department happen, and Howard Goodman for deciding to come here to MGH and being the chair for many years, and then Bob Kingston running the show in our department, and now Jeannie Lee. It's always been a very great place to do science, and we had this amazing support from Höchst, which was a very, very strange sponsorship. And we got to know the scientists involved. One of the first calls this morning at 5 a.m. was Ernst Winokur, who's been the champion of our department and a major figure in German science. And he was our connection to Höchst through his connection. And I've gotten to know him for 40 years, right? And these are wonderful connections that have. And Ann Klebanski, I contacted, 15 years ago saying, I have an idea about anorexia. And, and she works on this. And the chances of a physician listening to a nematode biologist's idea on anorexia was absolutely zero a priori. But she did. She did. And, that's a sign of a great place, right? That she was willing to do that. And we even did blood tests, not liver biopsies, but blood, But that I was totally impressed with that. So I'm very happy with my home at MGH, and it's always been great. What am I supposed to take questions? What am I supposed to do?
Right now, there are a couple of mics floating around, so just ask your question into the microphone so the live stream can pick it up, please.
Oh, and I need to thank my lab over the years. It adds up to about 100 people over a 40-year period, something like that. PhDs and postdocs and grad students. And, it's been phenomenal to work with you. And often, I'm not telling them where to go. They're telling me where they've been. And the joy of doing genetics, where you mutagenize the animal and ask what is the mutation that emerges from our genetic screen that we've done, is the surprises. There's 20,000 genes approximately, and I know something about 500, maybe 1,000 of them, but I don't know something about 20,000. And there's a constant, hey, look what came out of the screen. And it's like, well, what is that? And you got to do a PubMed and look at the homolog and see what the homolog does in some other organism. And the fact of the matter is, there's about 500,000 molecular biologists in the world now, and there's only 20,000 genes approximately, and that's spread out over every organism. So the chances that somebody has worked on a gene you bump into is quite high. But they don't know that you're working on it, and you don't know they're working on it. And so there's plenty of chances for surprises. And the surprises are what keep you young in science. And so I am constantly surprised, and my ignorance is bliss.
Question, you want to raise your hand?
Just say who you are.
Good morning and congratulations. I'm Brianna. I work with Channel 5, so I am not nearly as knowledgeable about this topic as the rest of the folks in here. But I would love to know, since your colleagues did speak about what a long-awaited moment this is for you, the feeling when you finally got the call or email or whoever shares this information with you, the feeling of accomplishment that you must have had. Can you describe what that moment was like after all these years?
Well, it was a realization. There are a lot of awards, but the Nobel is in its own class in terms of how much attention it gets. So I don't know, I've probably gotten 10 different awards over the last 20 years. There's never been a press conference like this, or TV cameras, or, nothing like this. So it's a completely different world when you're talking about that. And, but this is the second one in our department, right? Jack Szostak received the Nobel, was it 2008, I think it was. And, that was a huge deal. We were all super happy. We got to go to Stockholm to sort of partake of it, which was super fun. They know how to party. When you see John Sulston wearing 6-shooters and chaps at the party that night, right? With shooting at target, I mean, they know how to party.
Hi, my name is Victoria Rodriguez. I'm a doctor from Bogota, Colombia, and I just can't believe I'm here. So I am just starstruck. And now that I have the chance to ask you something, I'm currently 25 years old, and I would like to know, what would you tell yourself, your 25-year-old self? Like
my 25-year-old self. Well, I was in graduate school at that point. But, from like 21 to 24, I was a disappointment to anybody who was thinking about where my career might go. Because I lived in my van and planted trees in the Pacific Northwest and traveled in third-class buses to Tierra del Fuego for a year. And so, but then in Cochabamba, Bolivia, I was bored with travel and I went to the Bolivian American Friendship something or other. It's probably a CIA front. And they had Scientific American magazines, and I spent a day reading Scientific American, and it was really a good day. And I said, you know what, maybe I'll go to graduate school. And so I came to Harvard for graduate school, and that was 76. And that was sort of the moment when recombinant DNA was really just starting to take off. And it was obvious that that was a revolution. And one of the reasons why it was obvious that it was a revolution is I had studied physics before that, and as an undergrad, and one of the things you learn in physics is the quantum revolution, quantum mechanics. And that's really hard. The math is hard. But what's important is that it was a sea change, and you could see that it dramatically changes the science. And so I think I learned in my spinal cord, what a sea change is, and sort of wanted to be part of that. But again, advice to a 25-year-old, pay attention to revolutions, and enjoy them, because they don't happen that often, and they're works of art.
Hi, Gary. Al Powell from the Harvard Gazette. I wonder if you could take us back to the moment when you realized that this discovery was not just a quirk of a nematode, but rather something that had broad effects, and maybe comment a little bit about, did you think it would be as big as it is now?
Yeah, I mean, so there was an atomization of biology before genomics where, you could work on one organism and you wouldn't necessarily see how it relates to a second organism. And the genome projects that started to emerge in 1995, which again is like 10 years after I start here, that makes it trivial to generalize. So anything you find in one organism, you can rather instantly look in another organism. So I think when it became clear to me that this was larger than I had known, was our genetics revealed a second microRNA. So the first one comes out and is discovered by Victor Ambros in 1992, approximately, maybe 91. And then the second one in 1997 by Amy Pasquinelli and Frank Slack and Brenda Reinhardt in my lab. And we start looking at that microRNA, and it turns out writing grants, the tension of writing grants is good for you. And so there I was, as usual, nervous about how they're going to mess with me and imagine the four-letter words. And so I said, well, I know, I should look and see if it's in humans. And so I'm at home writing a grant, miserable. And this is in the days before we had internet at 100 megabyte, whatever. And it was a 9600 baud modem, right? From my study at home. That's how you send data, right? And I do a blast in to the human genome, which is not done. It's like 10% done, 20% done. And boom, it comes back matching the human genome. It's like, holy shit, this is great. And that was the sort of the indication that these are conserved. And, we run into this constantly in our little worms where people say, a nematode, it's not even a mammal, right? And it's a slimy, it's a worm that's this big. It's a millimeter long, If you want to know what a millimeter is, hold your finger a millimeter apart. And so, and you run into this at NIH, and we're at a hospital, right? And they were like, well, what about humans? What about patients? And so, but the genome projects have really helped that. And recently, we do a lot of comparisons between now we know a lot about all the genes that are in the microRNA pathway. And when we look at who has them and who doesn't, it turns out that mammals are missing a bunch of things that ancestral eukaryotes, that is the organisms with nuclei, the ancestors, all of us, had a very potent small RNA system, and for some stupid reason, mammals have dumped it. It must have to do with our brains. But if you look at the organisms that have the super potent, the animals that have the super potent small RNA system that worms have, it's scorpions and ticks and coral and clams. And it's the most badass organisms on the planet, right? And that makes our little worms badass. And no one ever thought to use that term for a worm. And so, we are asserting ourselves now. And I was asserting this before the Nobel stinking prize.
Hi, Gary. Congratulations. I don't have a question, but I just want to add to all the accolades that they were talked about you earlier, how wonderful you are as a person. I have spent a number of years as postdoc at Mol Bio back in the 90s, and I can tell to the people that they don't know you that you are an amazing person, a lot of fun to be around, and down to earth. And thank you for being who you are.
Thank you. Thank you.
Hi there, I'm Akshaya. I'm reporting for the Harvard Crimson. I would just like to hear a little bit more about kind of what Harvard and MGH means to you, how they've supported you, and kind of what it means to do research at a hospital.
Yeah, Harvard was super important to me in the sense that I went to grad school, at the biolabs with the rhinos, and that was sort of my home for, was it 5 years? And then after grad school, the Society of Fellows, which is a kind of a Harvard postdoctoral thing that's not just in biology, was very important. It's where I met my wife, Natasha Stoller. She was a junior fellow at the Society of Fellows. That's really an amazing organization, very anachronistic, little silver carts with cigars after dinner. It's not anymore, I don't think, but it did 40 years ago, and that caught my attention, But also, there were these mythics figures that were my professors, right? Just these unbelievably great scientists. And I always think that there's a sum of about, say, 25 just knock your socks off seminars that I heard as a grad student. And often I didn't know, it wasn't by a super famous person, but it was just somebody who was clearly magnificent. And you would see that, and that adds up to an education, right? And so I was exposed to cutting just great science over and over and over again. And then having friends in different fields, especially the Society of Fellows was very polyglot, right? So there were physicists and astronomers and historians, and that, you were really exposed to a university as it ought to be.
further questions from members of the media?
Good morning and congratulations. I'm Anil. I'm with Stat News. I have a question about what you think this award says about the value of basic science. You've touched on it a couple of times. And also to the point of credit, I know you don't choose who wins these awards, but do you think there was anyone else that maybe should have been named with you and your colleagues?
Oh, that's a good question. I mean, you could have come up, Nobel has this loony 3, the max of 3, and so that always messes with things. And and that excludes people if you add up to 4. And so that, there's a lot of that goes on. What was your other question? Oh, the basic research. Yeah. So the Nobel Committee is really good at basic, right? And I can't help but say that this year being discussed was Joel Habner and the GLP-1 work, which comes out of MGH. And how many institutions have 2 different groups of people up, vying for the Nobel Prize at one? That's amazing, right? And watching, because I went out to visit Joel last week. And so he's 90 years old almost. And I watched that work develop, and it's clearly at that status. And so it's amazing that MGH has that. That's more applied. That's more of the endocrinology that MGH really wanted out of our department, but boy, did they do it. And that's just an incredible story. I'm so proud of all of them.
No further questions? Thank you.
Okay.